All right, this is the third leg of our Genetics Problems Lab work through video. And we're going to start here with incomplete dominance. And remember, codominance is when you see both completely. Incomplete dominance is a mix or a midway between the two. So when the phenotype of the heterozygote is somewhere between those two of the possible two homozygotes, like in the example in lecture was a pink flower from a red and white parent flowers. And that's the example that's being used here as well. So you don't use a lowercase letter because neither is recessive. So you use an uppercase letter for both. And it's showing you here how you would represent homozygous red and homozygous white. So what's the color of the flowers with the genotype CRCW? Well, that would be midway in between. So the color in this case would be pink, just like in the example that we covered in lecture. So let's work through this practice problem. 2 short-tailed cats mated and had a litter of 11 kittens. 3 kittens with no tail, 6 with short tails, and 2 with long tails. How could you explain this variety of tail lengths amongst the litter of kittens? Well, again, we're seeing an example of 2 extremes, no tail and a long tail, and then something midway between the two. So that's how you know that you're looking at a situation of incomplete dominance. So to make a key, we'll use T for tail and N for no tail and L for long tail. And then that means that the heterozygote would have a short tail. So the parental cross in this instance, I would ask to give the genotypes for the 3 different phenotypes. Well, I'll just go ahead and add those over here to my key. A cross between 2 short-tailed cats. So 2 short-tailed cats, oops. It's going to be this genotype crossed with this genotype. That's an ugly N. Okay, so we are going to have 25% no tail. 25% long tail, and 50% short tail. All right, so now looking a little closer at the difference between codominance and incomplete dominance. And here's, I love these little Dr. Seuss examples. They're fun. So we're going to set up, it says practice setting up keys, genotypes, and phenotypes listed in each set. So remember that medium trait must always be heterozygous. All right, so birds can be blue, white, or white with blue-tipped feathers. So that would be the one, this would be the bluebird. This would be the white bird. And this would be the white with blue tips. And this is not a light blue. This is white with a blue tip. So this would be codominance. Flowers can be white, pink, or red. We've already done this one, but we'll do it again. And this one is in complete dominance because there's a mix between the two. A who can have curly hair, spiked hair, or a mix of both curly and spiked. So where this pink is a meet in the middle, but here you're seeing full white and full blue in different places, this is where you're also seeing full curly and full spiked. So this is codominance. And it's going to be, and I'm just choosing these letters, C for color, H for hair, curly. Oh, I forgot to put the colors here. I just noticed that. That's red, white, these are the phenotypes, and pink. All right, curly. And we'll do this for spiked. And this would be the mix of the two hairs. A sneetch can be tall, medium, or short. Short is midway between the two. Not a mix, but midway. And so that's incomplete dominance. And I'll use H again for height. So HT. Oh, I keep forgetting to put the phenotype. Would be tall. HS would be short. And HTHS would be medium. A bleak-so can be spotted, white, or black. Now, if it said gray, it would be incomplete dominance, but it doesn't say gray, it says spotted. So this is codominance because you're seeing both colors fully, just not all one or the other. So let's use C for coat, color, or for color. And this would be black. This one would be white, and this would be spotted. And then it says, from the examples A through in the above list, which letters correspond to codominant traits and which are incomplete? Well, codominant, A is codominant, C and E. And B and D are incomplete dominants. And again, codominant, if you've got one extreme is blue, the other extreme is white, the codominant, you're seeing full white and full blue, just in the same organism. So spotted. You're not seeing a midway point. In the example of incomplete dominance, you are seeing a midway point. Midway between tall and short is medium. Midway between white and red is pink. So again, the key here, because this can be confusing, is a midway point. And here you're seeing a mix of both. All right, then it gives you the next 2 pages, I think, are some practice problems, and the solutions are in the lab, but I'm going to work through these anyway so that if you had a hard time, having the solution doesn't do you any good if you still don't understand how you should have arrived at it. So I'm going to work through each of these. Hopefully you did take time to work through these on your own, and if you didn't, then stop the video here and try to work through at least the practice problems on your own so that you can see where you are. You don't want to wait until you get to the exam to find out you didn't really know how to work these, but you thought you did. The exam, even though you're going to be doing it from home, it will be timed. So if you don't know how to do these and you haven't really practiced doing them on your own, you won't have time to look up the answers or to figure out how to do it once you're in the exam. So it's still set up like a normal exam in that it will require you to put that time in on the front end. All right, so let's get started with these. A man who's a carrier for sickle cell anemia, a recessive genetic disease, has a child with a normal non-carrier woman. So this is recessive, it's not sex-linked. What proportion of their children is expected to be afflicted with sickle cell anemia? Well, let's look at this cross. Oops. Oh, that's right. This is where they like to use the N for the normal. All right, so this would be the key. And a man who is a carrier for sickle cell anemia, so the male genotype is this, has a child with a normal non-carrier woman. What proportion of their children is expected to be afflicted with sickle cell anemia? So you do a square. And we'll put her here. All right, so 50% are homozygous dominant and 50% are heterozygous. However, since this is a recessive mutation, 100% are normal. So the question, what proportion of their children are expected to be afflicted with sickle cell anemia? 0%. Hemophilia is an X-linked recessive disease that prevents blood clotting. If a woman who is a carrier for hemophilia has children with a normal man, what proportion of their sons is expected to have hemophilia? All right, so we know it's X-linked recessive. And the woman is the carrier, so she's going to be like this. A normal man, this is going to be his genotype. And I can't remember what the designation for the hemophilia example was, but you don't have to use the same designation every time as long as it's a correct example. There's more than one for every problem, and then you're able to get the answer. So N in this case would be normal, and little n would be bleeder. And they're asking what proportion of the sons. Well, let's do the cross. We can go ahead and do this portion, but these are the females, so they weren't actually part of this question. All right, so in this instance, the females would be 50% homozygous dominant for normal. The other 50% of the females would be heterozygous, so they would still be normal. For the males, 50% are going to be normal, and 50% are going to have the recessive allele and be bleeders or have hemophilia. All right, moving on. Neurofibromatosis is a genetic disorder associated with an allele of one autosomal gene. So autosomal means it's not a sex-linked. Individuals with neurofibromatosis have uneven skin pigmentation and skin tumors. A man with neurofibromatosis has children with a normal woman who does not carry the allele for this disorder. Okay, so she's normal and does not carry the allele. He has neurofibromatosis, and since we know well, we don't really know yet whether or not it's recessive or dominant, so I might have jumped the gun. But we know that she's homozygous something, because she's not even a carrier. And he has the disorder. A couple has 5 children, 3 of whom have neurofibromatosis, so that's a higher, more than half. the most likely explanation for this outcome is that neurofibromatosis is, I think the most likely outcome is that it is a dominant trait, but I'm not just going to circle that answer. I'm going to write it here for right now. And the reason why is because we know that she's not even a carrier, and yet still 3 out of 5 of their children have it. So let's practice that. Let's say she's not even a carrier. And in this case, the big N would be, that's a mouthful, neurofibromatosis. And the little n would be normal. And so in this case, he would be heterozygous. Oops, I didn't make him heterozygous. Sorry, if I could go back and delete just that part, I would, but I don't want to go back 20 minutes, so you just have to follow my correction of that. I just wrote down the phenotype wrong when I went to do that. Or the genotype. You got to make sure you get your genotypes right when you write them over here. All right, so 50%, in this case, 50% of the offspring would have neurofibromatosis. So that is possible. Because 3 out of 5, we can't get it even 50% to know for sure. Now let's just look at what would happen if it were recessive, so we can eliminate any wrong answers. So if it were a recessive trait, and we could also look at if it's dominant and he's homozygous. Let's do that quickly first. If it was dominant and he was homozygous, so then we'd be looking at this. What would happen here? If this were a big N instead, 100% of them would have it. So that's not it. So we can, if it's dominant, it's heterozygous, not homozygous. So now let's look at the possibility of it being recessive. And if it were recessive, then this would be his genotype, and hers, if she's not even a carrier, would have to be this, and you would end up in the same situation as the example I just did. If either parent is homozygous dominant and the other is homozygous recessive, all of them are going to end up heterozygous normal. So the best answer here is the first one that we tried, dominant, heterozygous, and so the answer here would be all right, moving on, looking at a pedigree. Now, we didn't have any of these yet in the lab, but I did do these in lecture. Consider the following pedigree for a human autosomal trait. So autosomal tells you that it's not on a sex chromosome. Note that in pedigrees, generations are indicated by Roman numerals, individuals, da, 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 da. Is the allele that determines this trait dominant or recessive? Well, if it were dominant, and either of these, see, in order for these two parents to have these 3 kids and the trait to show up, if it were dominant, one of them would have had to have shown it. And if it were dominant, they would have shown it. So I don't think it can be dominant. So we can go ahead and eliminate that. So the answer here is recessive, because in generation 1, there would have to be a dominant phenotype for it to show up in generation 2. All right, using symbols big A and little a for the dominant and recessive alleles respectively, what is the genotype of the affected male in the second generation? All right, so we've determined it's not dominant, so then this affected male would have to have this genotype, which so would the affected female here. And for this male to have a child that's also homozygous recessive, this female has to be heterozygous. Because that's the only way you're going to get this combination. If female 3-2, so they're saying this female, has children with a heterozygous man, what proportion of their children is expected to have this trait? So that's the cross, the same cross as her parents. That's going to be too small. I don't know if you can hear my cat purring. My cat is wanting to get involved in the lesson here. All right, and so you're going to have 50% with this genotype and 50% with the homozygous recessive genotype. Does it tell us what the trait is? No, but the percentage that would be expected to have the trait, which we know is recessive because these are the shaded individuals, so it would be 50%. Okay. I'm going to need to do a separate video for the last one because I am getting close to 30 minutes and that's where YouTube likes to make you pay to upload the video. So I'm going to stop here and we'll have one more video where I do the questions that you're looking at right now.